Hello, everybody. My name is Matt Farnitano. I am a fourth year grad student at the University of Georgia, and today I will be talking to you about ongoing introgression dynamics in a hybrid population of Mimulus monkeyflowers. So we know that hybridization has profound and varied impacts on species. These can range from adaptive introgression of particular alleles, creation of whole new hybrid lineages, collapse of a species through hybridization, or reinforcement of species differences through the selection against hybridization. And we want to know what conditions lead hybridization towards these particular outcomes, or can we predict the outcomes of hybridization from the conditions that we see? And today I'm mostly focused on adaptive introgression. So that would be the selective introduction of particular alleles from one population into another without bringing the whole rest of the genome with them. So how do we actually get adaptive introgression? Well, the first thing we need, obviously, is some kind of initial hybridization event that produces F1 or first generation hybrids. And this can be a rare event. It can be more common, but it has to at least be possible. And these hybrids have to be able to survive and reproduce. Once this happens, there's a few different possibilities for how things can go. In one possibility, this F1 hybrid mates back to one of the pure species, produces offspring that are more like the pure species, but still have some ancestry from the second species. And if this happens over and over again, you can end up with this backcrossing scenario where you get a small chunk of, say, the yellow species in a mostly red background. And this is probably the simplest case of how you would get adaptive introgression. But there's a few limitations to this. First of all, selection is a numbers game. So if you want to select only the best alleles and get rid of everything else, you're going to have to do this over and over and over again, so that selection has the numbers to be able to act efficiently. And you're, of course, also limited by the efficiency of recombination in breaking apart those yellow alleles, so that only the beneficial ones can be kept and the less beneficial ones can be removed by selection. So what if instead we had a situation where the original first generation hybrid either self-fertilizes or mates with another first generation hybrid and produces hybrid offspring? Those offspring then do the same thing, mating with other hybrids or self-fertilizing, and this happens for multiple generations. You end up with this hybrid swarm of lots of different individuals with hybrid ancestry, but they have different contributions of alleles from red and yellow. And in this scenario, we have a pool of lots of different hybrids to select from. Each hybrid has their own unique combination of red and yellow alleles, and we have lots of opportunity for recombination to break apart those alleles and allow selection to act on the best combinations of alleles. And then you can get back crossing from this hybrid swarm and more efficiently introgress only the alleles that are beneficial in the red background. But of course, there are some limitations to this scenario as well. You're limited by the fitness and persistence of those hybrids. If the hybrid swarm is really unfit and can't reproduce itself, then this is never going to happen. And you're also limited by the connectivity of the hybrid swarm back to the parental population. If this hybrid swarm is totally isolated, then you're never going to get the back crossing that you need to introgress only the beneficial alleles. So keeping those scenarios in mind, we're going to take a look at our study system, Mimulus guttatus and Mimulus nasutus. So Mimulus guttatus is an outcrossing wildflower. It's got big, showy yellow flowers. Mimulus nasutus is a close relative that is self-fertilizing. So it's got these tiny little yellow flowers. Sometimes they don't even open up. When they do, they're much smaller than guttatus, and it's mostly self-fertilizing. They have overlapping ranges, lots of opportunity to come into contact, and we know that at least in some places they do hybridize. So this data from 2012 This structure plot shows a cline of ancestry with hybrid individuals that have a majority Guttatus ancestry, but different contributions of Nasutus ancestry in the genome. And we want to look at this population in a little bit more detail and try to understand what's really going on. So first of all, we want to know, is there a stable, persistent population of hybrids? And if so, how is this population being maintained? Is there frequent production of new F1 hybrids every generation that's allowing the population to persist? Or are hybrids persisting by mating with other hybrids, creating this sort of hybrid swarm scenario? And then do hybrids mate with other parentals, or are they mostly mating with other hybrids? Is there connectivity between a hybrid swarm and the parental population? So to do this, we took samples from the field of maternal plants, and then we also collected seeds from those same maternal plants, grew those offspring in the greenhouse, and we did DNA extractions and Illumina sequencing for both the maternal plants and the paired offspring samples. And we used this low coverage Illumina sequencing approach, which has the benefit of sequencing lots of individuals at relatively low cost. But there is some missing data because when you sequence individuals at low coverage, you're going to miss out on certain genotypes for certain individuals. But we can use bioinformatic approaches to account for that missing data. So one approach is to use this angst suite of programs that uses genotype likelihoods rather than hard SNP calls, and then combines those likelihoods across many sites to get population level information. The other approach is this ancestry infer pipeline, which takes prior information about allele frequencies in the two parental populations and uses that with a hidden Markov model to assign ancestry across the genome for each individual, combining across multiple sites to account for that missing data problem. And it can assign ancestry as homozygous for species 1, heterozygous, or homozygous for species 2. So using this data, we can ask whether the composition of the hybrid population is stable across years. And we can compare our new data, which is from the 2019 and 2021 field seasons, to data from back in 2012. And we can see that, yes, there does appear to be a stable population across years. We see the same sort of cline of ancestry with majority Guttatus and some Nasutus ancestry, the same pattern of hybridization that we saw back in 2012. So we do have this stable, persistent population of hybrids, and we want to know, how is that hybrid population created or maintained? Do we see frequent production of new F1 hybrids? To do that, we want to look at the ancestry proportion of all the individuals in our sample, starting with just the maternal individuals. And first, what we see is there are no actual 50-50 individuals. And these ones that are close to 50-50, you can look at the heterozygosity versus homozygosity and tell that they are definitely not F1 hybrids. They're at least second or more generation. So we don't have any new F1 hybrids in our maternal samples, but do we have any in our offspring samples? To do that, I'm going to hide the hybrids just for a minute and look at our pure or mostly pure species individuals, Guttatus versus Nasutus, and plot the offspring onto this plot. And what we see is that the offspring pretty much match the maternal ancestry. If you were a Guttatus mom, you had Guttatus offspring. We don't see any F1 hybrids being created in this new data. So if that's the case, how is our hybrid population being maintained across years? Well, we can look at what's going on with the hybrids themselves and ask whether hybrids are mating with parentals or with other hybrids. And so if we plot the offspring of each of our hybrid maternal plants, we see that on average, they mostly have a hybrid ancestry, which you would expect. And to simplify this, I'm going to now plot just the mean or the average of each fruit from our data set. So this is the mean of multiple offspring from one fruit. And we start to see that for a lot of these fruits, we have a little bit of a skew towards more Guttatus-like ancestry than their mom, but we have some fruits that have a ancestry that really matches their mom closely. And we can take this one step further by extrapolating what we think the paternal genome contribution for each of those fruits is. So if we know the maternal ancestry, we know the average offspring ancestry, then we can plot what we think the paternal ancestry had to have been. And if that's plotted in these red triangles, what you see is most hybrid moms mated with hybrid dads. However, we do see some cases where a hybrid mom mated with what looks like a pure or mostly pure Guttatus dad. So we see a little bit of both. We see hybrids mating with hybrids. That's probably the majority of our data set, but also hybrids mating back to Mimulus guttatus. And taking a step back, it seems like this situation is kind of the perfect storm for adaptive introgression. We've got this hybrid swarm scenario with a persistent hybrid population that's maintained across years. Even though the production of new F1s is rare, hybrids mating with other hybrids are able to keep this population going across years. And even though hybrids mostly mate with other hybrids, there is some backcrossing that occurs that is allowing those alleles to infiltrate the Mimulus guttatus population with potentially very efficient selection and recombination to allow the adaptive alleles to make their way back into Mimulus guttatus. So at this point, you're probably asking, well, do you see adaptive introgression in this population? And I'm not going to answer that definitively just yet. I'm still working through the data, but I do have some intriguing new results that I'd like to show you. So if we take a look at just these mostly pure Mimulus guttatus moms, so about 30 individuals with less than 10% Nasutus ancestry, and we plot the allele frequency of Nasutus alleles within that population, what we see is kind of a crazy amount of variation. So the solid red line here is the mean, the average Nasutus ancestry in those individuals, and it's sitting about 2.5%, very low. But we see a wide variety where some parts of the genome have as much as 50% or more Nasutus ancestry. Are those alleles adaptive, and are they introgressing because they're adaptive? We don't know for sure yet, but it's certainly something that we would love to explore more and very intriguing. So at this point, I'd like to thank my advisor, Andrea Swigart, the other members of the Swigart Lab, former and current, my thesis committee, who's been very helpful, and especially our collaborator, Keith Carley, who's done a lot of the field work to make this project possible. Thank you very much for listening.